For decades, arthritis has been treated as an unavoidable part of aging. Once cartilage in our joints starts to wear down, doctors can only try to manage the pain, slow the damage, or ultimately replace the joint entirely. But what if damaged cartilage could actually be regenerated? What if aging joints could be restored instead of replaced? A new scientific breakthrough from Stanford Medicine suggests this may no longer be science fiction. Cartilage is the smooth, rubber-like tissue that cushions our joints and allows bones to move easily against each other. It's found in the knees, hips, shoulders, and many other joints throughout the body. The problem is that cartilage has almost no ability to heal itself. Unlike skin or bone, once cartilage is damaged, it usually keeps getting worse. Over time, this leads to osteoarthritis, the most common form of arthritis and a leading cause of chronic pain and disability worldwide. Current arthritis treatments don't fix the root problem. Painkillers reduce discomfort, anti-inflammatory drugs calm swelling, and physical therapy helps maintain movement. But none of these treatments restore lost cartilage. When damage becomes severe, joint replacement surgery is often the only option. This is why scientists have long searched for a way to regenerate cartilage itself. Now, researchers at Stanford Medicine believe they have found a key piece of that puzzle. Their discovery centers on a protein that increases in our bodies as we age. This protein is called 15-PGDH, and it plays a major role in how tissues age over time. As levels of this protein rise, the body's ability to repair and regenerate tissue declines. The researchers discovered that this protein actively suppresses cartilage regeneration. Simple terms, it tells cartilage cells to slow down, age faster, and stop repairing damage. The exciting part is what happens when this protein is blocked. Using a special drug that inhibits 15-PGDH, scientists treated older mice that had age-related cartilage loss. The results were remarkable. The mice began to regenerate cartilage that had already been lost. Even more impressive, the regenerated cartilage was high-quality hyaline cartilage, the same smooth, shock-absorbing cartilage found in healthy joints. The treated mice showed improved joint function, better mobility, and reduced signs of arthritis. Their joints behaved more like those of younger animals. This wasn't just slowing down damage, it was actual reversal of age-related cartilage degeneration. The researchers also tested this approach in mice with knee injuries that normally lead to osteoarthritis, similar to ACL injuries in humans. Mice that received the inhibitor were significantly less likely to develop arthritis compared to untreated mice. This suggests the therapy may not only reverse existing damage, but could also help prevent arthritis from developing after joint injuries. Perhaps even more encouraging, the scientists didn't stop at animal studies. They tested the same inhibitor on human cartilage samples taken from patients undergoing knee replacement surgery. These samples came from severely worn and damaged joints. When exposed to the inhibitor in laboratory conditions, the human cartilage cells began showing signs of regeneration. This is important because it demonstrates that the mechanism works in human tissue, not just in animals. While this is still early research, it strongly suggests that the same biological process could potentially be activated in human joints. What makes this discovery especially unique is that it doesn't rely on stem cell transplants or complex surgical procedures. Instead, it works by reprogramming the body's existing cartilage cells. By blocking the aging signal, these cells begin to behave more like younger cells, producing new cartilage and repairing damage naturally. This approach could be simpler, safer, and more scalable than many previous attempts at cartilage regeneration. It also opens the door to treatments that could be delivered through injections rather than surgery. Of course, there's an important reality check. This treatment has not yet been tested in large-scale human clinical trials for arthritis. More research is needed to confirm safety, effectiveness, and long-term outcomes. However, there is reason for optimism. A version of this inhibitor is already being studied in humans for age-related muscle weakness. If those trials confirm safety, it could accelerate the path toward human trials focused on cartilage regeneration. If successful, this therapy could fundamentally change how we treat arthritis. Instead of managing pain or replacing joints, doctors may one day be able to restore cartilage, preserve natural joints, and prevent arthritis from progressing in the first place. For millions of people suffering from joint pain, stiffness, and limited mobility, this research represents something incredibly rare in arthritis treatment: real hope. While we are still years away from a widely available treatment, the science is pointing toward a future where aging does not automatically mean joint degeneration. This discovery reminds us that aging is not just about wear and tear. It is also controlled by biological signals that, in some cases, can be adjusted or reversed. As research continues, cartilage regeneration may become one of the most powerful examples of how modern science can rewrite what we believe is possible for the human body. If this research continues to progress as expected, the question may soon change from how do we live with arthritis to how do we prevent and reverse it.